After the second session of our overall computational immunology session, we had to change the order of talks. I will present first, and then the two remaining speakers of that session before the coffee break will follow me. I'm very happy. It's my great pleasure to present to you our work on systems biology of T cells in cancer and infection. Our goal at Princeton, in my lab at Princeton University, is to address fundamental problems in biology and medicine by integrative analysis of multidimensional data. And among several things we are interested in the lab, we dedicate most of our effort to systems biology analysis of T cells or all kinds of immune cells across different contexts. We basically study regulatory genomics of immune cells. Why are we doing this? As you know, of course, cells in a multicellular organism are all very different in structure and function, and we would like to associate it with the gene expression profiles in order to better understand mechanistically all these differences. And we want, of course, to understand how gene expression is regulated. And we do this by analysis of publicly available and newly generated in the lab or with collaborators regulatory genomics data, such as RNA-seq, for example, for gene expression profiling, ATAC-seq for chromatin accessibility profiling, and therefore for assessing epigenomic and transcription factor binding state of the cell. And we also use, for example, chromatin interaction data, such as Hi-C. And it turns out that immune cells or immune systems and all these technologies that immunologists have developed are perfect, very highly suitable for studying fundamental aspects of regulatory genomics of mammalian cells in vivo in physiologically relevant contexts. But very importantly, collectively, all these experimental and computational technologies also help us understand fundamental questions about immune cell function and immune system with a wide range of clinical applications. We specifically focus on in our studies, in our work, we focus mostly on the T cells. And now I'm going to give you a little intro into what kind of concepts are needed to understand the rest of my talk. I will also present you our ongoing and then our past and ongoing work, and I will also try to emphasize computational challenges and some solutions and opportunities that arise while doing all this work. CD8 T cells are the central component of adaptive immunity. They are essential in defense against viral and bacterial infections and in tumor immunity. And normally, for example, in acute infection, they recognize the antigen and they respond and proliferate and develop so-called effector state, which actually kills the infection, and then eventually develop so-called memory, which enables more efficient secondary response to the pathogen. But in some circumstances, CD8 T cells develop so-called dysfunction, or also sometimes called exhaustion, somewhere during the process of, let's say, tumor initiation to premalignancy or to the establishment of a solid tumor, or during the progression of the infection, which eventually becomes chronic, T cells do not mount a functional effector response and they become dysfunctional. We wanted to perform systematic meta-analysis of genomic data for CD8 T cell dysfunction across different contexts, across different mouse models, in order to get a better molecular understanding of this concept of CD8 T cell dysfunction, which is a fundamental aspect of adaptive immunity, but also with a wide range of clinical applications in autoimmunity, in cancer, and infectious diseases. We want to understand dysfunction in order to be able to prevent, predict, reverse, or impose this state of T cell dysfunction. So we wanted to understand how T cell dysfunction is similar or different across different mouse models of chronic infection and tumors. We want to distinguish fundamental properties, molecular properties, from the model-specific effects, because ultimately, of course, we want to generalize all this to human. So we want to see how generalizable this is. We wanted to understand how T cells progress from naive to terminally dysfunctional state, and what are the underlying transcription factors and epigenetic factors that are underlying these processes. So we collected the compendium of bulk subpopulation ATAC-seq data from multiple different publications, and we pre-processed all this data in a uniform manner, and we constructed the atlas of chromatin accessibility peaks, which we think correspond to enhancers and promoters of gene expression. we, for example, here I'm showing you a snapshot of this data near the gene PD1. And you can see the peaks corresponding to promoters and enhancers on the top of this snapshot. Maybe a little hard to see, but we also collected data from multiple different functional states as they were characterized and profiled in each individual study based on the sorting strategy and the experimental setup of each individual study. So, for example, we have naive cells from multiple different studies. We have functional effector and memory cells from multiple different studies. And we have all kinds of dysfunctional cells from chronic infections and different tumors. And we needed to apply a certain generalized linear model to remove batch effect associated with the technical aspects of how the data was generated in each individual study. And after this batch effect correction, what we get is this picture where each point here corresponds to this 150,000 dimensional vector of chromatin accessibility values across the entire our atlas in each individual state. The color of the point corresponds to the functional state of the cell, again, as characterized in each individual study. And the shape of the symbol corresponds to the source of the data. So you can see that after batch effect correction, all the naive cells, for example, from multiple different studies, they cluster together. And functional effector and memory cells, broadly speaking, also cluster together in the middle of this PCA plot. But what's most remarkable is these dysfunctional cells also cluster together very tightly. And this includes T cells from chronic infections from multiple different studies, includes cells from multiple different tumor models, for example, hepatocarcinoma model, melanoma model. This includes cells both adoptively transferred CAR T cells and endogenous T cells. This also includes cells at different stages of, at different time points during the development of the tumors. All these cells cluster together, so we suggest that there exists this universal program of dysfunction which can be observed across different studies. And I think what's remarkable is that we can do this, we can make this observation based on the computational reanalysis of publicly available data. And we are now expanding this and are building compendium to expand to many other T cell functional states and T cell subsets. Now, with all this ataxic data, we are very interested in of course, epigenomic state and epigenomic regulation of the cells. So we can look, of course, at, and we want to look at the regulators of these states. We want to associate regulators, transcriptional regulators, with the functional states that we are observing. Of course, we can look at the overall chromatin, differential chromatin accessibility of the genes encoding transcription factors, for example, which we think are drivers of these processes. But we also can look at directly at targets of these transcription factors. And the idea is that if we have a peak of high accessibility, we think that maybe this corresponds to a promoter or an enhancer where a transcription factor binds, and this affects the expression of a nearby gene. And when the accessibility is lower or absent, we think that maybe this transcription factor doesn't have a way to function. And we can analyze this data together with our prior knowledge accumulated in the publicly available databases of the sequence-specific transcription factor binding. So we used all these motifs. This is the form in which all these transcription factor binding sequence specificities exist, to scan them through the entire atlas of chromatin accessibility peaks, and we have this kind of prior understanding of where these transcription factors could bind. But then we build a regularized negative binomial regression model to predict accessibility in each individual functional state via these transcription factor motif hits. And the inferred transcription factor coefficients in this model then represent for us the cell state specific transcription factor activity of in each individual functional state. So essentially what we are doing is we are mapping these 150,000 dimensional vectors of chromatin accessibility into something like 100 or so dimensional transcription factor activity space. So here's, for example, here are the results of this analysis. Columns correspond to the transcription, to the functional states, and the rows correspond to the transcription factor motifs. And you can see that, largely speaking, broadly for naive, functional, and dysfunctional cells, we recapitulate the relationship between cell states quite well. You can see this by looking at the color map of the functional annotations on the top of this plot. And then we can aggregate the information across functional, across replicates of the, or examples of the functional state within a study and across studies, and we then can prioritize or make hypotheses about transcription factor activity across different functional states. And we come up with known transcription factors and identify some new ones which could potentially be associated with functional states with this approach. Now, this was at the bulk and cell population level, but we are also very interested in zooming further into the heterogeneity of the CD8 T cell responses across different immune challenges. So we wanted to apply single cell analysis to address this. And for this, we use the standard model of lymphocytic choriomeningitis virus, where if you infect mice with the Armstrong clone of this virus, the mice develop the acute response and efficiently eliminate the infection. And this has been the standard model for the acute infection. And if mice are infected with so-called clone 13 of LCMV, they eventually develop so-called chronic infection, and they do not efficiently eliminate the virus. And this has been the standard model for studying T cell dysfunction. And in fact, some of the data that I showed you earlier in the compendium, in bulk ataxic compendium, actually comes from this exact model. So we generated single cell ataxic data at day 7 after the initiation of infection in these mice. And we did, of course, here I'm showing you the standard pre-processing, normalization, dimensionality reduction, clustering of the data. And I want to note that while for single cell RNA-seq, some of these very standard now questions are at least they're well established, or at least are well studied, and we know many steps what should be done. It turns out that for single cell ataxic data, it's much more challenging, and many of the questions that we take for granted, many of the solutions we take for granted now in single cell RNA-seq don't quite work or don't work at all for single cell ataxic. And this is, of course, because even more extreme sparsity of single cell ataxic data as compared with single cell RNA, and also different distributions that read counts follow for that data. But nevertheless, we have to proceed. So we did an analysis which we find reasonable, and then we found that we used also what it was helpful is also that we could compare this data with our bulk ataxic data compendium that we collected for multiple different cell populations. And collectively, after this analysis, we identified the cells on the left of this map as the cells probably which are not even responding to the infection, because we profiled the total splenic CD8 T cell population, including the cells which are not even specific for the virus. So that's what we are probably very clearly seeing as a combination of naive and memory cells from prior exposure to different kinds of pathogens on the left of the map. And we also have activated cells on the right side of the map. And we also see a range of different functional states that we are capturing, including some states which are much more clearly dysfunctional and are dominated by the cells which we profile in the chronic infection, or what would become chronic infection after the infection progresses, and also the subsets, the clusters which are dominated by the Armstrong clone and are probably functional cells. And then we can again apply our predictive model, this time at not at the bulk, but a pseudo-bulk level. And we actually recapitulate some of the transcription factors we found with the previous analysis, and we also kind of refine and zoom into some of the transcription factors probably associated with some of these functional states. And we are now working on generalizing this approach to make it truly single cell. But even at this level, it helps us, we think, to associate transcription factor activities with functional states. So this was a little bit about what are the approaches and challenges we faced in this previously published study. To summarize this study, we performed meta-analysis of chromatin accessibility and gene expression, actually, which I didn't show you, in T cells across different mouse models. And we found that these chronically activated T cells across different mouse models follow a common path towards dysfunction, probably addressing some of the controversy in the field about how similar or different these concepts of T cell dysfunction or exhaustion are between different models. And we use multiple different types of analysis to prioritize cell state specific transcription factors in the defined T cell trajectories. Now, what we are interested next in the lab is we are, again, interested very much in regulatory genomics of T cell functional states. We want to understand how do CD8 T cells commit to becoming functional or dysfunctional, what are the epigenetic and transcriptional regulatory problems and transcription factors, enhancers and repressors driving these processes, and how all these processes are affected with perturbations and treatments, including clinically relevant treatments, such as, for example, anti-PD-1 immunotherapy. And we are very interested in how these mechanisms are evolutionarily conserved between mouse and human, or in other words, very practically, how much of this information we are learning in mice can be actually used to guide our analysis of similar data in humans. And of course, we are interested in going beyond individual cell types, and we want to study how cells interact with each other and how cell-cell interactions and spatial localization regulate different kinds of T-cell activation and functional states. And to give you just a snapshot of the data we are currently generating and we have generated and we are in early stages of analysis is, for example, we have generated the multi-omic single-cell RNA plus attack data in the same model of LCMV infection in order to better understand and zoom into where this commitment to dysfunction is happening, or functional state or function. And one more aspect of the data that we're experimental setup that we are leveraging was already mentioned a little bit and described a little bit by Sarah in Sarah's talk. We are doing our genomic analysis in the F1 hybrid cross between two different mouse strains, which gives us a great opportunity to leverage this genetic heterogeneity and use it as a kind of a natural experimental system to associate allele-specific transcription factor binding with allele-specific accessibility and allele-specific gene expression. We are seeing a lot of different functional states. We decided also to go after both CD4 and CD8 T cells because there is a lot of interest now in profiling not only CD8 but CD4 T cells as well in this context, and seeing how not only kind of main suspects but helper CD4 T cells might also be different between different contexts of T cell activation stimulation. But this also presents further challenges. And despite the fact that immunologists will always tell you that CD4 and CD8 T cells, especially at this stage in the spleen, are already well differentiated from each other transcriptionally and epigenomically using RNA and attack, we see them sometimes very close to each other. And it's hard sometimes for us to even define which cells are CD4 and CD8 without having specific markers for that. So this is one set of challenges, but we know that allele-specific analysis, which we have high hopes for, already works at a single cell level, or at least at the pseudobulk level, despite the sparsity. We have published portion of that data recently, and we demonstrated that allele-specific analysis can help us further zoom into transcription factors, which are cell state specific. And we are also very interested in kind of going higher order aspects of chromatin interactions and epigenomic organization, and we are now profiling, I think, the data which hasn't been published yet at this level of resolution of high C in different subsets of T cells, and we are seeing some very interesting results at different levels of resolution in that data. So with that, I want to thank my lab, my, of course, mentor, Christina Leslie, who is also going to speak later today, my postdoc mentor, and now also my new lab, which I have established at Princeton University. We have a great team. We are recruiting, and we are very open to collaborations. And we have many interesting things ahead of us. Thank you very much for your attention. I'm happy to take any of your questions. I can take the questions myself.
Hi, thanks for a great talk. I was just wondering what your understanding is of T cell dysfunction as being related to this kind of balance between either infection or tumor control, and then on the other hand, autoimmunity, and how potentially kind of disrupting or eventually drugging or something like that, this process of dysfunction to shift it could potentially affect this, risk of autoimmunity and those potential effects.
Yeah, I think it's a very good question. And ultimately, of course, as with anything, we want to address this with the data. And in order to really see how similar or different T cell states are between infections and tumors, but also autoimmunity, we need to build a bigger compendia and bigger data sets. We are definitely very interested in that, but I can hardly address your question right now before doing this analysis. But people have observed both similarities and differences and have generated already at a single cell level and of course at the bulk cell population level, all kinds of data sets that, I mean, at least RNA-seq and ATAC-seq that I have talked about. So we have at least something to already try to combine, and we're working on this, or we have plans of working on this, of course.
Hi, amazing talk. I'm Avi from New York Genome Center. Again, the idea of using bulk to figure out the annotation in the single cell is pretty cool. As we already seen that unsupervised clustering and then annotation is difficult just based on the single cell 8x data set. So I was wondering, have you tried like single cell references, like based on which were developed based on single cell 8x data set and then transfer back to your data set, and instead of using bulk as a reference, use single cell 8x data set as a reference, where like I think single cell ssci, which have like millions of cells and they have very good annotation, maybe you can use that to transfer back and the annotation would be much more refined. But
that's a great question. So for the study I presented, we started it a while ago, several years ago, and at that time I don't think these resources existed. So now maybe we should if we were to do this from scratch, we will probably do something like this. That said, many of these data sets often are profiling, and this is, I mean, this is a general comment about the challenges we have with anything related with T cells. All these single cell analysis methodology works great, and there are many different ideas that are being published, but very often they are tested on the data sets which have a lot of heterogeneity, let's say, in expression or chromatin accessibility. We work with T cells where we think we have much more subtle differences between different functional subsets of cells. Okay, so some of these, I'm looking forward to seeing these resources that you mentioned, which maybe capture this very well. But often what we see is that these general approaches, let's say, at profiling all kinds of cells, like let's say all circulating immune cells or all tumor cells, they sometimes have a hard time to really carefully annotate subsets of, let's say, T cells. And also they will have less of a consistency or agreement between different studies which do this. So I think while these resources are great and we should all, have one big resource which will allow us to analyze, T cells data in any other new context, I think we are not there yet. But definitely we should use that.